indeed, in my talk, I'm focusing on those next steps because what is interesting at this day is to consider that, okay, we have the proof of concept. We know that it's a possibility. Now, what is missing? And what is missing is actually a long list of things. That is what we need to transfer these proof of concepts into a real therapy, which would be applicable to millions of patients, because that's the goal of the substitutive cell therapy, is first a question of cost, because what we do is expensive. It's expensive because we are working in labs, because we don't care much about reducing the cost. But even if we care, it's a lot of cultural media, of cytokines, of plastic, of time for technicians and equipment, and so on. And we need to focus on that, because indeed there is no way we can treat millions of patients with treatments that would cost $1 million or more, which it's not exactly the case now, but may reach those levels because of quality and so on. So the first thing is really cost killing, and cost killing means changes in the way we manufacture the cells, because we won't be able to reduce the cost sufficiently. We may reduce by 10%, 20%, 30%, but it will not be sufficient to actually go through to the market. So first thing is that. Second thing is quantity, because for instance, to make epidermis, just a 10 centimeter square of epidermis, we need 3 million cells produced in the lab. But 3 million plus what we need for quality controls, for loss, for whatever, let's say 5 million. 5 million, if we are dealing, for instance, with skin ulceration related to diabetes type 2, it's millions of people. So you multiply 5 million by millions. And then the problem is, how do you produce those cells? Because they need weeks of culture, and you can't do that by hand. That is, it's something that will require automation, that will require industry to actually get into the field. And again, because also you don't want cost to explode, because you are producing those cells. And it's not only it's not the only question of quantity. It's also a question of how you deliver the cells, who will do it, where, and who you will train to do these kind of things, and so on. So it's an entire field that has to be built, in which researchers at this moment are frontline, but industry has to take over, and then the hospitals and the clinicians have to get into the field, and nurses and so on. And so it's really an entire new world that has to be built with in mind this idea of quantity. And then you have quality. And quality, in our case, means two things. When you introduce cells into the body of someone, and I would say allogenic cells, because if you go to cells that you want to produce from the patient itself, and then retransplant after correcting or changing things and so on, then you get into cost, which is just impossible to solve. It takes months, and so you can't do that. So you have to work on allogenic cells. But allogenic cells will produce, most of them, an immune response. And what we do at this moment with the clinical trial is that we put patients on immunosuppressants. But we can't do that for millions of patients because it requires a lot of care, because it will create a lot of adverse effects, and it may kill people because the immunosuppressive treatments are very harsh treatments. And so we need to find another way around the problem. And a lot of labs are working at this moment in order to find ways to create cell lines that are stealth cell lines for the immune system. And that will be one of the next steps, unavoidably. That is, we need that. And conversely, we need to protect the patient against the cells. Just if the cells go wild, if what you have transplanted start being burgeoning or creating problems in the patient, you have to get a way to retrieve them, to withdraw them. And that's not something that you can do easily, because the cells will be immersed into the tissue, and you can't just take off a piece of the brain or the retina of the patient and say you get rid of the cells that way. So we have to engineer the cells so that we can actually kill them from the outside by an exogenous means when we need to. So it's a lot of questions, and questions that require answers that are in the hands of the scientists. That is, it's really still now, even though we are talking about progression between the lab to the industry and to the clinic, all those questions require scientific answers, and the field will progress on that basis.